Hello, can you see? Hello, can you see about anything about the picture? You can see here. Just let us see the picture and let's guess what the picture is about. You can see here very small particles. I think you can guess. It's the picture about something small, smaller or the smallest. If you guess like this, I think you have already understood what I am going to talk about. Yes, it is a picture of a virus and its gamma phase, an example of a virus. I guess now we are going to talk about virus and virology. This is virology, and what we are going to read from free Wikipedia, the free encyclopedia. Now let's see what virology is. Virology is the scientific study of viruses, submicroscopic parasitic organisms of genetic materials contained in a protein coat and a virus-like agents. It focuses on the following aspects of viruses: their structure, classification and evolution, their ways to infect and exploit host cells for reproduction, their interaction with host organism. Physiology and immunity, the diseases they cause, the techniques to isolate and culture them, and their use in research and therapy. Virology is a subfield of microbiology. Virology is a subfield of microbiology. You can see the picture here of microbiology. Okay. The identification of the causative agent of tobacco mosaic disease as a novel pathogen by Martinus Beijerinck, 1898. You can see the picture of Martinus Beijerinck is now acknowledged as the official beginning of the field of virology as a discipline, distinct from bacteriology. He realized the source was neither a bacterial nor a fungal infection, but something completely different. Beijerinck used the word virus to describe the mysterious agent in his contagium vivum fluidum, that means contagious living fluid. Now we are going to talk about virus structure and classification, viral disease and host defenses, micromolecular biology, research and viral therapy, sequencing of viruses, other uses of viruses, history, see also referencing and further reading and external links. Virus structure and classification. A major branch of virology is virus classification. Virus can be classified according to the host cell they infect. Animal viruses, plant viruses, fungal viruses and bacteriophages, viruses infecting bacteria, which include the most complex viruses. Another classification uses the geometric shape of their capsid, often a helix or an icosahedron, or the virus's structure, e.g. the presence or absence of a lipid envelope. Viruses range in size from about 30 nanometer to about 450 nanometer, which means that most of them cannot be seen with light microscopes. This is the picture of a light microscope. The shape and structure of viruses has been studied by electron microscopy. NMR spectroscopy. This is NMR spectroscopy. And X-ray crystallography. This is the picture of X-ray crystallography. The most useful and most widely used classification system distinguishes viruses according to the type of nucleic acid. This is the picture of nucleic acid they use as genetic material and the viral replication. Viral replication method they employ to coax host cells into producing more viruses. DNA viruses divided into double-stranded DNA viruses and single-stranded DNA viruses. RNA viruses divided into positive sense single-stranded RNA viruses, negative sense single-stranded RNA viruses, including retroviruses. Virology also studies subviral particles, infectious entities notably smaller and simpler than viruses. Viroids, naked circular RNA, molecules infecting plants, satellites. Nucleic acid molecules with or without a capsid that require a helper virus for infection and reproduction. And prions, proteins that can exist in a pathological conformation that induces other prion molecules to assume that same conformation. Taxa, taxa in virology are not necessarily monophyletic. As the evolutionary relationships of the various virus groups remain unclear, three hypotheses regarding their origin exist. One, viruses arose from non-living matter separately from yet in parallel to cells, perhaps in the form of self-replicating RNA ribosomes similar to viroids. Two, Viruses arose by genome reduction from earlier, more competent cellular life forms that became parasites to host cells and subsequently lost most of their functional. Examples of such tiny parasitic prokaryotes are mycoplasma and nanoarchaea. 3. Virus arose from mobile genetic elements of cells such as transposons, retrotransposons or plasmids that became encapsulated in protein capsids, acquired the ability to break free from the host cell and infect other cells. Of particular interest there is mimivirus, a giant virus that infects amoebae and encodes much of the molecular machinery traditionally associated with bacteria. Two possibilities are that it is a simplified version of a parasitic prokaryote, or it originated as a simpler virus that acquired genes from its host. The evolution of viruses, which often occurs in concert with the evolution of their host, is studied in the field of viral evolution. While viruses reproduce and evolve, they do not engage in metabolism, do not move, depend on a host cell for reproduction. The often debated question of whether they are alive or not is a matter of definition that does not affect the biological reality of viruses. Viral diseases and virus host defenses. But now we are going to end our class about virus, because I hope you have got a lot of information by doing this, and I'd like to thank you for being with viruses. And once again I'd like to tell you that we got information from here about virology. Virology is the scientific study of viruses. Scientific study, you can see the picture of scientific study here. I hope you have got a lot of information, and thank you for subscribing and sharing the information. I hope you enjoyed reading on virology and the scientific method. Thank you very much.